A deep sea TV.
Do we have continued interest in this battle royale, or would you like to move on?
Maybe just another 30 seconds.
Oh, you got it.
See what
Here comes
this
another.
New one does when he comes in.
I feel like this is one of those like MMA fights, or not MMA, the wrestling, WWE, where you just have people running from the audience.
Huh.
That definitely must be some kind of tissue there. The new worm that just joined is trying to get in on it as well.
Oh, yeah.
Yeah, now you can see the cellophane-like end of the tube on the one in the upper left.
Uncomfortable.
Yeah, it's just interesting whether the ones that have more sediment on them, if they are older. Or
That's what I was wondering, if it was a life stage kind of thing.
You got another guy coming in.
What is going on?
Because it almost seems to be part of their tube. It's because even when the worms crawl on top of each other, it doesn't fall off. But maybe that's just they have some sort of mucus.
Tube pile. And
all right, I guess we've seen most of the action we'll see here.
what?
All right, I guess video, you can call it. If you want to toss in the towel and bring this fight complete.
Just wondering if the guy on the right is going to escape.
Another question that came in the chat room about how we knew that there were seeps in this area, and whether that came from previous dives or from maps of the seafloor. So we've had a few times that there,
looks like the fish came out a little bit more from up above.
There has been imaging of the water column, so the seeps produce bubbles, and the bubbles go up into the water column, and our instruments, our acoustic instruments, can detect the contrast between the gas and the water. And so the plumes are found typically before the seep. The seep is the seafloor feature is found. And the kind of work we do today is to verify
pretty much only seen the head so far, right?
Seepage at the seafloor feeding the water column plumes. So often now it's sort of the geophysics of looking at the plumes in the water column that tells us there's a seep there. In this particular case, these seeps were identified prior to 2014 based on some data collected by the Okeanos Explorer. Subsequently, the U.S. Geological Survey has done a lot of surveys, including in this area, and found a lot more seeps and verified these ones. And then other people at the U.S. Geological Survey and their colleagues, including some of the people listening today, did a previous dive here and verified that there was, in fact, seafloor, there was seafloor that sort of corresponded to the fact that there would be seepage here. So we have a lot of ways, but we're getting much better at using remote sensing to find these features instead of having to dive every single place and look for the needle in a haystack.
All right.
That was a great answer,
Yeah,
Carolyn.
great summary. Thanks. We're going to do a quick pilot change.
No problem.
And Bernie Ball just gave us just sent a note here that the clear area at the anterior ends of the tubes have an internal double or triple valve system.
I've been running around 10. If you get closer, come up maybe to 12 or something.
I didn't
but
know that at all. I don't know if it's to channel water deeper into the tube or to keep out sediment or.
All right, so I think the additional mats were basically dead ahead, right?
Okay. The southern hake to just bury most of their body underneath the sediment.
Cool jellyfish, right?
We've seen so many of them this expedition.
Might be a siphonophore.
Hmm?
Is that a siphonophore?
I guess Bernie noticed the valves opening when the worms were moving, so maybe it is to allow water to flow into the tube. I didn't notice that, but I was looking at what they were eating in their heads really more than the tube.
Okay, 225 is the.
That's the next waypoint, but
next waypoint.
If you see mats.
I see it went off the starboard there.
Understood.
I think we gotta look at those, right?
Another very active hagfish in its typical swimming mode, scavenging across the seafloor.
Piles of worms.
Just pile.
Looks like the game of pickup sticks. You know, you have to like chop. Is that what it's called? Pickup sticks.
What I find crazy is that they all take so long to move. How do they all get moved up like that?
They're pretty fast. I mean,
something must smell good. You know, like.
I think there's dead fish on the ground. They're all eaten or something.
Yeah. Come on in, partial. Dust
so
clear,
we're seeing
okay.
some of the
Frame
midwater
this,
organisms again,
bubbling out.
suggested shrimps and that barracuda that just passed. And we've seen a lot of salps today.
A lot today.
Yeah, not much there. Mosey over to the next one.
Oh, there's another group of the tube worms, I mean the quill worms here.
Feeding pile.
Okay, come on in again. Just frame it. I already don't see any bubbles or anything, but see if there's anything else interesting. Scanning. Yeah.
Wow, that's an amazing zoom.
Mm-hmm. That bacterial mat has a little sediment cover to it, so it's probably not as recent.
Okay, moving on to the next patch.
The one on the upper left looks much more white and less sedimented.
Yeah. So is that newer or more active?
Looks like there's a lot of these groups of the quill worms.
Yeah, we should take a couple more zooms on them, see what they're eating.
Which group are you looking for?
I'm curious to know if the snails are being attacked by the tube worms or vice versa.
Right.
I think the tube worms can outrun the snail, actually, so I don't know about a snail sneaking up on a tube worm.
We'll take a quick zoom here.
There have been several associated with each of those piles.
This one has I thought that was a hole, but maybe it was just the eel pout.
Definitely not the concentration we saw downslope of the bacterial mats.
Okay, we can go over to this pile over here.
Most of the landscape was covered with them.
See if we can see what they're going after.
Can we do a quick zoom on that pile of quill worms up there?
Yep, headed there now. Caught my eye. There's a lot of little traces. See that?
No, stationary. You need to move?
Stand by a video. I'm just getting really close in a sort of top-down view so you can see what they're feeding on. It might be a
their
shrimp.
Tracks. And there's another fish in the burrow.
Coming down. Settling. On it. Yeah.
That's quite a pile of tube worms, huh?
Come on in. See what they've got. Shrimp, maybe?
Lunchtime.
No, another snail.
Oh, a squid.
That is a squid.
Wow.
They're feeding on a squid.
Look at the tentacle.
Oh, the poor squid.
Save me.
Nightmares tonight.
As long as those worms stay in those nice straight tubes, they're not going to bother me too much.
I'm not watching this.
It's when they're out of their body, out of their tube, I don't like.
They have such beautiful colors when they're out, though. Like little rainbow.
We'll probably have to take the sample out of its tube to get the DNA sample.
What do you mean, we? You're the biologist. I'll be washing sediments.
In the other corner of the well,
yes.
far away.
Far away.
Good day to be a geologist.
It is. I'm happy.
So what you're saying is you want to
Although
scoop
I had
up
to
all dozen
pluck all
of these
the
worms.
worms out of the sediment yesterday, and they did not have
What
tubes.
bearings you want, co-pilot?
Whatever bearing you're going on.
Okay. You want 20 meters or 30?
So these guys are one of the top predators down here, it seems.
Apparently. Who would have
Bridges
thought?
are we now?
How are we feeling, video?
Of
Good.
course, we
Wanna
also
move
saw
on?
that
Sure.
fish in
I'll
the
get
hole.
get the
that
lasers
just
on it.
down from our image.
Yeah, I'd like
We
to
saw
move
one of those
20
eat a
meters,
squid
bearing
right
225
water column
degrees,
before
speed decimal
also.
2, same as last.
Okay, that's probably
It's
good.
a good copy.
Look at all the trail
Want a
marks.
quick look at the
Yeah,
fish.
let's take a peek at him.
Yeah, sounds
now
good.
that
Come on
is,
And then we have an anatomy
that's
right
the
there.
rye mouth.
That is a beautiful fish.
AKA grumpy fish.
Yes.
What's it actually called?
Yeah, Cheryl said we were watching one up close and a squid came by and both the fish and the squid disappeared down the hole very quickly.
And in the replay video, you can see that the squid got sucked into the mouth of the fish.
Yep.
Well,
I wonder if they eat quillworms. If these
well,
fish eat
copepods.
quillworms. If so, they would not be hungry.
Is it a copod or copop?
The snail might wander in
Copo?
to that burrow
It's a
accidentally.
race to the burrow. The snail is winning.
Uh-oh.
Oh!
Wow. That scared me.
Look at the snail peeking down saying, what just happened? Oh my gosh.
Save.
There was a fish there a second ago.
Save. Oh, no!
Oh my goodness. The most action ever.
Dum, dum, dum. Oh my
All
gosh.
right.
Wow, that was some action. We were just talking about in the chat room whether that was a barracuda.
Hello, this is what's going on.
Chris, how are you?
You just missed some
Yeah.
action.
Did you see that?
I've been watching the video. I just thought I would chime in and give you a name on those worms. I don't know a lot about them, but we see them in shallow water videos a lot.
Hold on, I
Those are
can't hear
members
you
of the
over
family
the,
Onuphidae.
Uh-huh.
They're watching it over the okay.
So they're called onuphids, and even in the shallow water species that I've seen, they do that strange carrying their worm tube around behavior. And it says that they're scavengers, not necessarily carnivores. So I don't know if they're feeding on that squid as a matter of predation. I would gather, based on shallow water species, that they're probably just taking advantage of the protein. So I don't know much about their general biology beyond that, but I just thought that would be useful information. And it also gives me a chance to plug Polychaete Day, hashtag Polychaete Day tomorrow on Twitter and elsewhere, which is in honor of Christian Foucault, who used to be the Smithsonian's curator of polychaetes. And so July 1st is his birthday. He passed away several years ago, but the worm community, the polychaete worm community, still honors his contributions by having this big day. And on Monday at the museum, we will actually have worms and other polychaete type of critters out for people to come see if you're here in Washington.
Well, that's great. Thanks for that info, Chris.
Sorry to plug, but
oh no, it's great.
It's educational, so there we are. But Anufids, so there we are.
Thanks a lot.
Okay, looking for the
I'll
next
talk to you later if
And
there's starfish.
everybody enjoy Polychaete Day tomorrow, and if you're in Washington, D.C., head to the Smithsonian on Monday.
Or you can visit us this evening in our wet lab as we pull out a polychaete from
Come in partial.
one of these tubes. We collected a quillworm earlier.
Still sneaking in. Can hang tight there.
And we are watching replays of that fish-eating fish event. It's quite dramatic.
I don't see much going on here.
Yeah, Amanda Demopoulos had just asked me if that was a barracuda, and I said yes, and then that one
hop
came
over to the next
flying
one.
into.
Yes.
He actually looked a little rough,
He
too.
does. Yeah, his skin is all mottled, and he might have been on his way down anyway, but he's definitely gone at this point. A lay and wait predator.
Seems like the best way to do it around here. Low energy. I want to see a two-worm snag a squid.
Move complete. Thanks, Bridge.
Scott said that we don't have enough days on the calendar to support all the diversity we see.
That's for sure.
There's something over there. Guess that's my next stop. Yeah, it might be a big... Yeah, no,
It's
yeah.
a mat.
It's a weird shaped mat. Sort of fish-like looking.
Yeah.
This
Yeah.
one looks like it's kind of
Gave
floating.
it a fake
It
shadow.
does.
I'm sure that's an
Yeah,
illusion.
Yeah.
it
Yeah.
is. It looks like shadow instead of the dark sediment just on the side.
Coming
Scott
down a
France
little
wants
bit.
to know when Bamboo Coral Day is. I think we should just choose a day, Scott.
Well,
Go
what's
ahead.
your birthday, Scott? We will establish your birthday before you pass.
Yes.
We won't wait.
Come on in. That's good.
So I guess those are just reduced
Yeah.
sediments on the bottom side of our...
Yeah, I'm
Ship
wondering
is,
where they came from,
stationary.
but it might have been just from one of these torpedoing squids coming in and scraping along and disturbing the sediment. Definitely looks like it's been disrupted.
Just kind of mosey over to the next one. It's right here. See if there's any action there.
It looks like it's that snail.
All sorts of animal tracks on these surfaces.
It looked like it's a snail that's in the lower left.
That large snail that we just passed.
Oh, yeah.
Yeah, if you follow the track, you can see it
Yep,
coming
that's
right
it.
off.
Oh, yeah, yep. So it might have been digging underneath there. Yeah, thanks, Bob.
Starting to get the swarm.
Yeah, can we take a quick zoom of the snail after you see this one?
Yeah, sure. Getting a little
Dusty.
swarm and dust here, but we can try for
Yeah,
it.
we've seen several of these today.
Coming down some. Stand by a second for me, video.
Sorry, I asked too late. And there's a good size comparison of the two species.
We're just come wide, let this dust clear a
We
little
collected
bit.
what
Get
we
dive
think
bombed by
is
all
the
these
larger of the two
critters
early
again.
on, but it wasn't nearly that large, so we'll know when we see the sample later today.
So when I get a chance, I'm going to look up this quillworm paper that was published in 2016 that Bernie Ball
Okay, this
has
might be the
pointed
best
out.
it's gonna get.
Um,
Yes.
with
Come on
field
in.
notes on charismatic and abundant epifaunal
What
polychaetes
is
of
this?
the U.S. Atlantic continental margin.
Stab their heads in the ground for a minute, and then
Yeah, I think they're just a little disoriented.
I've never
from
really
us.
thought of a polychaete as charismatic.
Well, that's why you need to read the paper.
Exactly. I need to get a new level of appreciation.
I'm definitely getting a better level, more appreciative of these organisms. Yeah, we've seen this snail mostly submerged in the sediments with only its foot sticking out.
I think it's time to mosey on. We're not going to get...
See that it's covered by it. Oh, there's another dead fish.
Dead? Yeah,
Nope,
I think he's
not
just,
dead.
I think they're just
I'm not dead yet.
a little dazed.
He's not dead, he's just resting.
It's like they like your lights and they just keep stirring up dust for you or something.
Oh yeah, I'm gonna push out past all these fish and krill and stuff, see if I can't get
those
some time without them. Though they are beautiful, they are also annoying. That one's just straight up bearing
And
its
Amanda
head.
Demopoulos from USGS said that they saw loads of them.
Come in, Marshall.
Dives around Baltimore and Norfolk
never know
Canyon.
what he's doing.
And quite
It's the ostrich
collected
fish.
quite a few for food web studies.
It thinks that if it buries its head,
their
you can't see it.
2017 Baltimore Canyon paper in MEPS found them in the adjacent slope in abundance as well. And here we have another burying hagfish.
Looks like he's made a couple burrows in the same area. You can see where he was earlier.
Is there something down there that he's going for, or is he just hiding?
Look at that. So I think they dig down at first and then they move sideways because they start sliding in much more rapidly.
Yeah, and there might be a tiny head of one poking up behind where this guy is going in.
Oh, is that a hagfish poking out there?
That makes sense because then they can
Try
sense
and get a
what
snap
is
on that.
moving around close to them on the sediment
Getting all
surface
those critters
and dart
back.
out when
Go quick before it
they
dusts.
think it's something to eat.
Yeah, see how much the sediment to his left, or to our left of him, has now bulged outward.
Hanging tight, yeah.
Yeah, can you drop down where the lasers are? Just to the left of the hagfish?
Huh? Sure we can. There's going to be a lot of dust and fish there, so I'm not sure what you're looking for is going to still be visible, but we can try. What are we going
his
for?
His head is poking out there.
That crack lower right. Come back.
Yeah, just letting it clear
Yep.
out. And I'm going to back up a little bit so I can get some tilt
Yeah,
back.
he's definitely disrupted the sediments to his side. That's fine.
Yeah, I don't
Pilot,
see his
if
head.
you want to keep moving.
I don't see him poking out at all. He's just lifted it up. Great. Mav is giving us our next direction
He's
ahead.
completely gone now.
Actually, there's one up here. Go for that.
Did we have someone just come online? On the phone?
Yeah, okay, you can come in on this mat, see if there's anything of interest. I guess it's going to be a dusty couple of minutes here.
We've had as many as 22 scientists, maybe even more, join us on a Saturday in the chat room. That's pretty darn good.
Yeah,
so
thank
we
you for
appreciate
taking
your time and contributions to improving our knowledge of this area.
Pushing out to the next one.
Ready for move?
Yeah.
What bearing you want? I was going 225, but looks like you're looking maybe.
And we are now at
I'll call it in
425
at 200.
meters. We've
Scanning along here.
We've scaled about 100
Bridge RV
meters
now.
of vertical relief.
Yeah, I'd like a move.
over this
Range
dive
20 meters, bearing 200, 200 degrees. Speed decimal
Sure.
2, all 2.
Off we go.
That's correct.
All right. Next up, another biomat. This is sort of like what they were dealing with this morning, huh? What's this little curlicue?
And the interaction of the midwater fauna with the seafloor here has kept this dive very
Come on in real quick.
entertaining.
It's a coral eel pout
Lots
or
to
something
look at.
else. Interesting.
Yep.
No.
Oh, that one's not doing so well.
No, he isn't.
I think he's
The
just
little
taking a
eel
nap.
pout.
Okay, we'll let him go.
I think he's upside down even.
He is.
He's not happy with the current situation. We'll leave him alone.
No.
Oh, I can gather a guess as to why that is, judging by the screen in front of me. I could venture a guess.
Look at like every single one of your lights is just this pool of.
Okay, well,
krill.
do I have room to give a push here? I do.
Pilots, this is watch one.
Just gonna get out of here real quick. Yes, go ahead, watch one.
Would you be able to introduce yourselves?
Absolutely.
Great, thanks.
Currently commanding this dust storm in the pilot chair is pilot Jeffrey Lanning. Sitting to my left,
Levi
navigator.
Levi Unuma.
Lars Murphy on co-pilot.
Over on the right side, Bob not on video.
And in the back, the clipping chair is Roland Bryan doing the clipping. We're all from the Global Foundation for Ocean Exploration. And to my left.
Hi, I'm Leslie Sauter from the College of Charleston in South Carolina, and I am the geology lead for this expedition.
And I am Cheryl Morrison
this
from
crab
the
yet?
U.S. Geological Survey Leetown Science Center in
No,
West
I'm not sure
Virginia.
which direction I'm going to go yet.
And
I don't have
I'm
another
the
map
biology
in view.
lead for the expedition.
I'm going to scan,
And
see if
next
I
to
can
me
catch
is
one
Derek
in the
Sowers,
cam
who's
or something
busy
and head that
mapping,
way.
and that is his lead. He's the mapping lead for this expedition. Doesn't have his microphone on, so
Okay, so
we'll just
chime in if you
introduce
see
him.
one.
He's already scouting out tomorrow's dive site.
And tomorrow will be our last dive. Dive 17.
Up there?
It's hard to believe that we've coming to the end.
Right,
It went by
lasers.
so fast.
It was a slow start, though.
Yeah, that's
We're stuck in port a few days, so we lost time there, and then we only had 2 sites that we had to relocate. Well, a few we had to relocate, but we only lost 1 dive day. And then there were a couple sites that we were chased by the Gulf Stream eastward, so we had to keep figuring out a new place because it was just too much current. And then another site, I guess it was the Pamlico Deep site. We couldn't dive because the surface currents and winds and waves, everything was causing a mess for the ship to be able to maintain position adequately, so we lost that site.
Yeah, we can take a partial. It doesn't seem like any activities here, though.
Fairly close to shore off of Cape Hatteras area and Cape Fear.
Good.
And we've been looking at canyons, different portions of different canyons.
Yeah.
And these sites are all very different from the earlier sites when we were farther offshore and in much deeper waters.
Okay, take off. Try to keep ahead of the storm.
Different distances or relations to the Gulf Stream location. So it's been fascinating to see the variations among all the different
straight
sites.
ahead too. This might be the move. If you want to follow me over.
Not only in the biota, but in the geology and sediment types and types of rocks. Haven't seen many in the last several days.
Yeah, not much going on there. Sorry for the quick zooms. We're just trying to stay ahead of that storm of krill if there's nothing pressing to look at.
We haven't seen many stony corals either, which was one of the objectives of the dive, was to find habitat, hard substrate habitat for coral populations and sponges as well.
Yeah, we've seen in work a little further north in the mid-Atlantic canyons, we've found extensive areas of corals on the steeper walls of the canyons, both corals and sponges. But so far with our dives in these canyons that are further south, we've only seen sedimented, highly sedimented, very steep slopes and ridges. But then we've also seen evidence of these seep areas where
Sled
gases are
distance
seeping out of the
312.
seafloor. And that was one of the goals of today's
Did we
dive,
pass
was
5
to
already?
verify
That's the side they
whether or
want to
not
follow
the
Matt's. I'm going to verify with them first.
plume that we were able to see in remotely
Didn't wanna
sensed data was actually
Cut them off
a,
mid-sentence.
Yeah.
whether we could put our eyes on it and see where it was coming out of
Come
the
on in.
seafloor. So these bacterial mats are a sign of that type of environment, and we've seen some bubbles today and extensive areas of these bacterial mats. And then some of the mats don't have bubbles associated with them at
Okay.
all.
Can move on.
Watch the, this is your nav speaking. So if you look at high pack, we're
There's
basically
one over
headed
here and the
directly to
rest
waypoint 6,
the
following the
south.
mats. I do notice you have a waypoint 5 over here. Is that a hard target that we need to scooch over to get, or would you rather follow the mats to 6?
And the
Understood.
last several dives have had
We'll do
very
that. Thank you.
similar types of substrate. This soft sediment
All right, Pats, we're following the mats to
doesn't
6.
show too much
that.
variability in it. There are not a lot of ripples or bed form
Seems like
features
it's going straight
indicating
uphill, which is great.
low velocity currents. The grain size is very fine, again, supporting the low velocity current environment. But what we have seen is completely different dominating biota, and including the fish.
Okay, come on out.
A few days ago, we saw brittle stars covering the surface. The next dive, we saw these pout eels, or eel pouts, excuse me. And I'm trying to think of what invertebrates we saw that were dominating. Today, we definitely are seeing the quill worms as the dominant invertebrate fauna here. Epifauna, certainly.
Yeah. Go for this guy, see how it is.
And all these areas have a tremendous amount of organic material being delivered to the seafloor as well. So there's plenty of food. It doesn't look like a coral reef environment. It's not densely populated with high diversity in a small area, but
Yeah, come on
there's
in,
plenty
video.
of food for a wide variety of organisms. They're just distributed more evenly across the landscape.
It's more like a two-dimensional habitat instead of
That's
three-dimensional.
right, exactly, yeah. And we don't see as
Something
many
out?
filter feeders here.
Right.
Because there isn't that current.
Sneaking in.
And probably too much sedimentation
here.
and nothing to attach to.
Yep.
Yeah. Okay.
And even on the very steep slopes, these sediments are cohesive enough so that they don't just tumble down the slopes. They stick together enough so that they are pretty much in place, except for the very surface that is still very water rich.
Understood.
And when organisms, through time, a lot of erosion does occur.
Bridge is Darwin,
over
yeah.
hundreds and
Yeah,
thousands
they can
of
move
years of that type
20
of
meters. That's
mini
20
avalanche
meters.
activity. You get
Bearing 210 degrees.
peaked
speed
ridges
decimal 2.
forming
Kicked up a little dust
from
here.
erosion of the sides.
My fault.
It's an interesting shrimp.
Yeah.
That's different from, oh, that, no, it is the kind that has the long tentacles that we saw buried in the sediment earlier.
Yeah.
Yeah. That's where my
He's about to be buried in the
down.
sediment.
You're going to help him along a little
Yep,
bit.
yep. We know he doesn't mind.
Some squid ink.
Yeah, I don't think we're going to track him. We can head up to the next.
Maybe now that we've seen that shrimp above the surface, somebody can help us identify it. Even
I
though
don't
we
know. We saw those
kind of
before.
lost
I'm not
it in
sure
the
what that is.
sandstorm or the mud storm.
tornado.
That's a big burrow.
Want to see if there's anybody home?
Sure.
Okay. That's right underneath me.
Looks filled in.
Yeah, I don't see a hole to it. Okay. Alright, scanning for the next mat. That one doesn't look like there's a hole associated. Try to push up current of all this dirt. See if I can find a mat. Yeah, I see it.
Ship's underway.
Copy.
Yesterday we encountered a miniature cliff. We zoomed in so tight we thought we were looking at some significant feature, but turned out it was only a few centimeters high, but it was just covered with pycnogonids, there were snails, there were worms. It was an environment that we saw several small octopuses too, and
come on in.
It was really interesting because even though it wasn't rock, it was more solidified sediments, not totally solidified. They fell apart when we nudged them, but it was the relatively hard substrate for the area, and so a whole community of organisms took advantage of it. So certainly not what we think of as hard bottom or hard ground, but organisms thought it was hard enough for them to attach to.
And one of those organisms was a white soft coral, and when we took one sample of it to try to get a really good identification of it, and when we got it out of the bio box, for one thing, it was very tiny, maybe about 3 or 4 centimeters, more like 3 centimeters.
It's about an inch long, yeah.
Yeah, and it was actually attached to a mud tube, so it wasn't even a hard, really hard substrate that it attached to. It was a mud tube. So I have a feeling that's why there weren't a lot of other corals in that area. That's definitely on the fringes of what a coral could handle.
There's a couple of them there.
And you've mentioned remotely sensed data using the multi-beam. The intensity of the acoustic signals return gives us an idea of how hard or soft the substrate
go ahead and make your heading change.
And
understood.
You know, if there's a mud and a compacted mud, it will show up as relatively soft and hard, and we may mistake that for rock because we think of hard substrate as rock. So we have to ground truth or verify those types of data as well. We make a map of the surface. We know what the seafloor depths are from multi-beam, and we also get a sense of what the character of the seabed is based on the intensity of that acoustic return. And but we don't really always know what that means, and it can fool us, and we're trying to learn more and more with that technology.
Yeah, and how to interpret
it. It's very important to be able to see what the substrate is.
It's a very large patch of dark sediment on the left there.
Yeah, I was going to snap a grab here. We can go for that dark patch if you prefer.
Sure, I mean we can hit one then the other. It doesn't matter which order.
So presumably a new venting surface would not have the bacterial mats right away. Is that likely that they haven't grown yet? So you can see the quillworm paths very distinctly in this kind of sediment. And that could just be from somebody burrowing and digging up the dark material, and the current has spread it out.
An idea of what we're looking at here.
Here's another snipe eel.
Looks like it's had a dinner lately.
Mm-hmm.
So this all is like
that is such fine grain material.
Fine grain material out of this, what looks like a hole here.
I wonder what made the hole.
Well, we're sneaking closer
It's so
and looking
fine.
down to try to find
I'm
out.
surprised. You know, it's not in clumps like we usually see from material that's been excavated.
Mm-hmm.
Understood. 160.
I don't think I'd stick my hand down there.
No.
Even in the name of science? Okay. I think we can go ahead and chase some biomats. I don't see anything hiding out.
Wow, look at all those quill worms.
Yeah, I see that. Looks good. Hot on the trail. Can start impartial.
Copy that. 160. All right. You guys need to move, or you guys feel comfortable? Understood.
Yeah. Just whatever's good for you guys. Yeah, we're nice and shallow. Okay, I think we got time here. Moving on. The next patch.
We keep seeing the patches of bacterial mats, but we haven't seen any active bubbling since that large, extensive area, which is a while ago now. This might be another one of the fish in the burrows. It's getting a little dusting.
And another cancer crab. That's the dominant crab in these parts. We haven't seen. Have we seen any squat lobsters today?
Yeah, we saw a few at
Early
the
on.
beginning. Yep.
Hoping that dust would clear so I can get a shot, but I don't see bubbles.
Yeah.
We can move to the next one.
Can you move straight up where the black sediment is coming out?
Yep.
It might be what's causing that drape of dark sediment that we saw below.
Start in video.
Yeah, it looked like it was just happening.
Yeah, it didn't look like a burrow. Just coming out. Where was it coming from?
Right under that worm, I guess.
Looked like there were two sources, the upper one and the lower.
None of that was there before.
When we first sat down, yeah.
Yeah, it definitely just happened.
Yeah, we're looking at it on the
There it comes
replay.
again.
There were two sites of effluence.
See the separation crack there?
Just very short-lived.
Yeah.
So.
I think there's something under there.
Yeah,
Yep.
you see there's a little crack in the sediments.
Yeah.
Maybe this is like a hagfish just buried itself.
Alive?
Well, I mean, we've seen a lot of hagfish, but it's a bit of a coincidence that almost at the same time black stuff came out of where we're looking now and where we just were. And Yeah, I don't know.
I think he's pretty stretched out.
And the next bump up has that kind of...
30 meters?
And a lot that's flowing
20.
downhill.
Okay. 210, good. Okay.
Pilot, can we zoom in on the little mound that
Bridge
is
ROV now.
about 2 o'clock?
With black on top.
Yeah, I'd like to move range to
Top
0
corner
meters.
of the screen right now?
Bearing 210
top
degrees,
corner.
speed decimal 2. So same as the last move we did.
And these all have
Good guy.
that. Just different degrees of darkness. Probably the more recent are black. I suppose it could be from a burrowing organism that brings all the material up at once and throws it up and it's caught in the current. But it certainly it didn't look like shoveling, one after the other.
There's a hagfish up. Oh, no, that's a snail. Sorry.
Be nice to sample, but it would all blow away, I'm afraid. It would look like the other sediment sample.
You need to stay 4 miles away.
These are tiny snails here that we're seeing.
So, yeah. A warship. Stay 4 miles away. It must be a Navy ship.
Yeah, I saw that.
All right, well, let's keep wandering upward.
There's something in this hole, maybe. Moving around.
Keep an eye
Shadow
out on
of a shrimp.
Yeah.
Shadow of a shrimp.
Keep an eye out on the horizon. It seems to happen just
Come
before
on
we get there.
Well, I wonder if the ROV touching down a little bit is enough to release a little bit of gas if there's...
Well, we also have all these critters darting around, bashing into the ground.
Yeah.
So their disturbances could be doing it as well.
Yep.
Seems like it's pretty far away from us. Upper left or upper right? Yeah, okay. Got eyes on it. Or a fish.
Quite a few worms here. For this entire dive.
Yeah.
A bed of worms.
Ooh. Team 4 nice and close.
One fish, dead fish, dead fish, hagfish. No,
No.
a big joke.
Have we seen one of these yet today?
I don't think so.
All right. Yeah, let's go on partial.
I think we saw one of these yesterday. Have to
Okay.
figure out what it is again.
Oh, he doesn't want to play. Thanks. Copy.
And that's a great shot of him. And I don't have a note about that one from yesterday.
No, I'll come back. All right, come on out, video. Start to catch up to this train. There's a dark patch.
Yeah, another dark
Big one of
patch.
those dark
Yeah.
areas.
Just doing a quick scan for any biomat. We've sort of lost the trail of those, or they've ended, I should say.
Okay.
So I'm just going to scan
There's
around.
fish in the
There's one.
barrel.
There's a fish in the burrow. There's also a biomat over here.
Yep.
So, that's good, at least. Can maybe get back on track this way. And there's one top right, one up top left. Got a couple more. Ooh, is that a tonguefish or something?
Oh yeah, tonguefish. We've only seen these on one dive so far.
Yeah.
Move complete. Thanks, Bridge.
Sneaking in on it.
And maybe Martha Nazinski is watching, and her husband Tom Monroe could give us an ID.
Yeah. No.
Oh my goodness. Thank you, co-pilot.
That snail better watch out.
Yeah.
It's got the fish and the quillworm watching it. Uh-oh. I don't think fish eat gastropods, though.
It's like a tough shell
It
for
would
a
be a
fish that
crunchy
size.
meal, wouldn't it? When these fish are younger, they swim like most fish, and they have an eye on each side of the duck.
Oh.
There's another one. Boy, I guess if we just wait long enough. I guess having two eyes on one side didn't help him very much.
What's that?
Oh my gosh, he's gone.
Yeah.
Well,
That's
I
over.
was going to say that as they approach maturity and begin to settle to a benthic lifestyle, one of the eyes migrates to the other side, so they have both eyes pointing upward, which is usually a good defense.
This is what's been diving into the bottom, I think, lately.
Yep.
And that's a barracuda.
All right, he's in a dust storm. We'll let him go. Yeah.
And here's
Go ahead,
another
Bridge.
bacterial mat with dark, really dark sediment.
Understood. 170 will be your new heading. Understood. Thanks. What magnitude does the DP read? I understand that current magnitude on the DP is a sum of all unknown forces, but what does it read? Decimal 8279. Thanks.
James Cameron would love this dive.
Yeah.
To make another sci-fi movie.
Okay. This mat is pretty much empty of activity. You can come out wide and mosey along.
The dark truth about seeps is what Tina Moldsova suggested as a title.
Making a heading change, pilots to 10 degrees.
Copy. Yeah,
Little did they know.
that shrimp's completely buried other than its head in 10A. Okay, there's another mat. Just scanning around to see if there's...
Looks like we're kind of on the top of this ridge now. Or at least
Understood. Thanks.
more slope ahead, but we're on the local high.
It's much broader than yesterday's. We had both slopes in one view. yesterday. Very narrow ridge crest and very steeply sloped on each side. It was really impressive.
It's kind of mosey in the direction we were going. Hopefully,
But it was all mud, I have to say.
Mm-hmm.
Well, this would probably be a good time to say something about the fact that we typically find carbonates, so certain kinds of rocks that form due to chemical reactions in association with methane. And so these usually form just below the seafloor or even deeper in the system, but then are sort of exposed by erosion. And so those are very common and provide hard substrate for a lot of chemosynthetic organisms. What's been interesting about the places where we found methane seepage here and in other places recently is that we haven't yet found the carbonates. So there could be carbonates below the surface, but the fact that none of them have been sort of exposed at the seafloor may partially explain why we're not seeing a richer diversity of chemosynthetic organisms here.
Right, without the hard substrate to attach to.
That's interesting. Thanks, Carolyn.
I wonder if there are carbonates below the surface that we can't see buried in sediment.
Okay.
Well, that's how they generally form, and that's one of the misconceptions. People think they form
seen this yet. We can
right
take a quick
on the
zoom.
seafloor or above the seafloor. The fact that we don't know, obviously, if they're down there, but obviously this is also an area with very high sedimentation rate. So it may be that we don't have everything we need to form them, or that the sedimentation rate is much higher so that they're being buried rapidly.
Yeah. Here's a ceryanthid tube anemone, and it looks like he's eating an amphipod, or trying to. An amphipod is stuck in his tentacles. And they have lots of sticky cells, and the amphipod is trying desperately to get away from those cells. Oops, and it might not work this time. He's being pushed in towards the anemone's mouth there.
Absolutely. 210 still good?
We saw something similar yesterday, but the amphipod got away and then got
Bridge is RV now.
snapped up by a crab.
Yeah,
that was sitting in the
down.
Just waiting for the
20
cast
meters.
off.
Trying to
Cast
get
off.
Bearing 210
Trying to keep it still,
degrees,
but
Taking the
speed decimal
leftovers.
2.
Fortunately, there's not enough force
Oh,
for
there
me
he
to fight
is.
against. It's just bouncing between my
Good copy.
thrusters being on and off. There we go.
Definitely wants to keep holding on to it.
Mm-hmm.
I can't tell if that's a tube in the back behind him or...
Yeah, maybe when we're done watching the anemone's lunch, we can pan over to the right a little bit on that tube.
Sure. Give me more zoom.
Oh, it looks like a quillworm, doesn't it?
I wonder if he wound up in a burrow and then the burrow got buried or something.
Yeah, he's standing up. It'd be hard to get out of that one.
You probably can't reach the floor like how he is. Oh.
And Tina said that this tube anemone that we're looking at might be
just up there.
Pachycerianthus borealis.
Around.